I have a bad feeling about this.
To understand thalassemia, we have to know that we have two major forms of hemoglobin. Hemoglobin F, which is composed of two alpha chains and two gamma chains, is the major form of hemoglobin in fetus. And adult person has hemoglobin A, which is composed of two alpha chains and two beta chains. The major pathological feature of alpha thalassemia is decrease in synthesis of the alpha-globin chains. The synthesis of alpha-globin chains decrease due to the deletion of genes that encode alpha-globin, and these genes are located on chromosome 16. And to explain this, we have to recall how hemoglobin is produced.
What does that got to do with anything?
Everything!
Here we have two chromosomes 16. Each chromosome 16 has two genes that contain information how to make alpha-globin chains. And this genetic information in two alpha-globin genes we use to produce alpha-globin chains. And then, after alpha-globin chains are produced, we use them to form hemoglobin molecules. In adult person, hemoglobin molecule is composed of two beta chains and two alpha chains. And together they form a proper hemoglobin A molecule, which is the major form of hemoglobin in adult person. So let's suppose that we have two hemoglobin molecules, and after these hemoglobin molecules are formed, we pack them into red blood cells, and inside the red blood cells, hemoglobin molecules carry oxygen to peripheral tissues.
How embarrassing! How embarrassing!
Now, there are a few possibilities how gene deletion can occur. It can be cis deletion or trans deletion. This feature we will need later. For now, we have to know that normally we have 4 genes on 2 separate chromosomes, and all of them encode alpha-globin chains, and we write this in this pattern. The first possibility is deletion of only one alpha-globin gene. This form of thalassemia we call α-thalassemia minima, and in this case, person will be totally asymptomatic. So let's explain this. The deletion of only one gene can be perfectly compensated by 3 other working genes. So in this case, it will cause just slight decrease in production of alpha-globin chains, which won't substantially affect hemoglobin structure and it won't affect hemoglobin concentration in the blood. So, person with thalassemia minima will have minimal decrease in hemoglobin production, which still will be within normal margins. And because of this, thalassemia minima is asymptomatic. The second possibility is deletion of 2 genes. And in this case, it can be trans deletion or it can be cis deletion. Deletion of 2 alpha genes we call thalassemia minor, and this form of thalassemia will cause mild microcytic anemia.
This is getting out of hand. Now there are 2 of them.
Also, we have to know that this form is worse because it can affect a child or a patient with thalassemia. So, if deletion of 2 genes occurs on both chromosomes, we call this trans deletion, which is better for a future child. And if gene deletion occurs on a single chromosome, we call this cis deletion, which has worse prognosis for a future child or a patient. So, with deletion of 2 genes, the production of alpha-globin chains becomes substantially affected, simply because 2 out of 4 engines are not working here. In this case, the structure of hemoglobin remains the same, but the concentration of hemoglobin in the blood will decrease, because we cannot make enough hemoglobin with such low production of alpha-globin chains, and in this case, mild anemia will develop. As compensatory response, bone marrow will try to make more red blood cells. So this will cause increase in red blood cells precursors, which are reticulocytes. So if thalassemia causes anemia, one of the diagnostic features can be increase in reticulocyte count. The third possibility is deletion of 3 genes. This form of thalassemia we call hemoglobin H disease. Hemoglobin H is a hemoglobin which is composed of 4 beta-globin chains, which is totally pathological. And hemoglobin H disease will cause moderate or severe microcytic anemia. So let's explain how these hemoglobin H molecules are formed. With deletion of 3 genes, the production of alpha-globin chains becomes very low.
We so die in here.
Just relax, we're not in trouble yet.
What yet? Monsters out there, leaking in here, all sinking and no power? When are you so thinking we are in trouble?
I mean, we have just one engine left. What can go wrong? As you see, alpha-globin chain is connected to beta-globin chain, and in normal condition the connection is strong. But not in this case. The problem is that with such dramatic decrease in alpha-globin chains production, the force between alpha and beta chains becomes extremely weak. Basically, in this case, there is such a small amount of alpha-globin chains that beta-globin molecules cannot even bind to them. And absence of alpha-globin chains force beta chains to bind to each other. This results in production of beta chains tetramers. And hemoglobin, which is formed of beta chain tetramers, called hemoglobin H. The problem with hemoglobin H is that, first of all, this hemoglobin cannot carry oxygen as hemoglobin A. But more important is that hemoglobin H is toxic for red blood cells. Accumulation of hemoglobin H inside the red blood cells, with time, will cause red blood cells destruction, which we call hemolysis. And hemolysis will cause decrease in red blood cell count and severe anemia. On this image, we can see the inclusions inside the red blood cell, which are in fact hemoglobin H molecules. But in clinical practice, the more reliable diagnostic method to determine hemoglobin H is electrophoresis.
What is it?
On this image, we can see an electrophoresis of a patient with hemoglobin H disease that has 3 deleted alpha chain genes. On the left, we can see chromatography, which determined hemoglobin H fraction, and on the right, we can see electrophoresis. As we can see, the major fraction of hemoglobin is hemoglobin A, and it's the reason why this patient is alive. But there is also hemoglobin H fraction, which tells us about the presence of beta chain tetramers, which is pathological. On this image, we can see blood analysis together with electrophoresis of a patient with thalassemia. Here we can see hemoglobin H fraction, and in this case there is 32% of hemoglobin H in the blood, which is a lot. So it's definitely a more severe case than previous one. In addition to this, we can see Hemoglobin Barts fraction, and this type of hemoglobin we will discuss later. But we already know that such pathological types of hemoglobin as hemoglobin H and hemoglobin Barts are toxic for red blood cells, and their accumulation provokes hemolysis. In this case, most probably, in response to hemolysis, bone marrow overproduces red blood cells. But the problem is that even if the quantity of red blood cells will be normal, the amount of hemoglobin inside the red blood cells will be low, because we cannot make a normal hemoglobin without alpha chains. And in this case, hemoglobin level is 83, which we classify as moderate anemia. And MCV is 64, so in this case anemia is microcytic. So if patient has decrease in hemoglobin production or increase in hemolysis, as a compensatory reaction, bone marrow begin to produce more red blood cells. And one of the precursors of red blood cells are reticulocytes. And in blood analysis in patients with thalassemia, we can often see increase in reticulocyte count. So in thalassemia, often, even despite hemolysis, red blood cell count is normal or even high. It is important because in iron deficiency anemia, red blood cell count is typically low, and the signature feature of thalassemia is the presence of target cells.
You saved my again.
What's this?
A local. Let's get out of here before more droids show up.
More?
On this image, we can see target cells, and we have to know that the second name for target cells are podocytes. Here we can compare target cells with normal cells. As we see, target cells in the center have increased intensity, which is caused by local accumulation of hemoglobin. And normally, hemoglobin is distributed in the peripheral regions of red blood cells. And the last possibility is deletion of all 4 alpha-globin chain genes. And this disorder we call Hemoglobin Barts disease. Hemoglobin Barts is a hemoglobin which is composed of 4 gamma-globin chains, and this disorder is absolutely lethal. A person with Hemoglobin Barts disease dies at fetal stage due to a condition known as Hydrops fetalis. So, Hemoglobin Barts disease can be determined only in fetus. There is no possibility how adult person can have deleted 4 alpha-globin genes. So let's explain this.
How can this be?
Recall that fetus has fetal hemoglobin, which is composed of 2 alpha chains and 2 gamma chains. So instead of beta chains, fetus has gamma chains, and in fetus, hemoglobin F is the major form of hemoglobin. So if deletion of all 4 alpha genes occurs, this causes complete disruption of alpha-globin chain production. And without alpha-globin chains, gamma-globin chains have no option but to bind to each other. This results in formation of gamma-chain tetramers, and hemoglobin that is composed of gamma-chain tetramers we call Hemoglobin Barts. The problem with Hemoglobin Barts is that this type of hemoglobin is extremely toxic for red blood cells. As a result, hemolysis of red blood cell occurs, and massive hemolysis of red blood cells in fetus manifests as Hydrops fetalis, which is absolutely lethal condition. So, on this image, we can see blood analysis and electrophoresis of a patient with Hydrops fetalis. In this case, patient had deletion of all 4 alpha-globin genes, which cause formation of Hemoglobin Barts, which is in fact 4 gamma-chain tetramers. As we see, in this case, red blood cell count is low. The reason is that Hemoglobin Barts is so toxic for red blood cells that it causes so massive hemolysis that it cannot be compensated by increase in red blood cells production. This results in decrease in red blood cell count and decrease in hemoglobin. As we see, hemoglobin level here is 64, which is extremely low. MCV is 97, which is normocytic. This feature is also one of the indicators that hemolysis in Hemoglobin Barts disease is a predominant pathogenic factor, because normocytic anemia is a feature of hemolytic anemia. And on electrophoresis, we can see a huge fraction of Hemoglobin Barts, and 95% of Hemoglobin Barts is absolutely lethal. So in this case, Hydrops fetalis developed and fetus died.
Another happy landing.
May the Force be with you.